Hello, everyone. Thank you so much for joining us today for the BioBuilder Showcase for Cohort 4 of our Idea Accelerator program. We have some really fantastic presentations from the JR Masterman School, and I'm so excited to get to present some of the students' work with you here today.
Hello, my name is Maria Karakousis, and I'm representing the Masterman Nano Dragons in presenting our project idea, Solving Microplastic Pollution Through Algae. We are Blue Dragons attending JR Masterman Public High School in the beautiful city of brotherly love. Our overall system design that we seek to pursue this year is biologically engineering a strain of algae so that it can indicate through bioluminescence when there are harmful levels of microplastics present in aquatic bodies. Our input for this system would be microparticles, including microplastics from plastic materials, particularly polyethylene and polypropylene, as well as microfibers from synthetic clothing, as these are extremely dangerous to aquatic ecosystems. Our output for the system would be the green fluorescent protein, GFP, in conjunction with ethylene and propylene. Some questions that we have as we prepare to embark on pursuing this idea this year include how we would effectively contain the rapid growth of green algae to mitigate the spread of harmful algal blooms, as those are also harmful to aquatic ecosystems. Also, which specific strain of green filamentous algae should we use as our chassis? And finally, how would the enzymes be incorporated into the algae to break down the microplastics? Thank you so much, and we are very excited to pursue this project this year.
Here we figured we still wanted to do something that relates to the environment because that's something we're all very passionate about. But we thought of making it perhaps broader than like an asbestos issue. And so we just thought about environmental problems that are plaguing the world today, and we thought of microplastics, which is a pretty big one. We just want to say thank you very much for holding this program. We really did enjoy working on this project together, and we look forward to the rest of the year where we hopefully get to implement some of what we planned.
Hello, my name is Justin Liu, and I am representing the Masterman Nano Dragons and presenting our project idea for the BioBuilder's Idea Accelerator program, curing Alzheimer's through white blood cells. Our overall system design is to send in modified white blood cells to clear beta amyloid plaques in the neural system. Inputs into such a project would include the modified cells and the plaquing in the brain. The outputs will be a remade neural system without these plaques. A question that we have is how would we be able to get a blood cell to make it through the blood-brain barrier? Another question we have is which enzymes will we utilize to break down the amyloid plaques, and how would we incorporate them into a white blood cell? A third question we have is how would we prevent the white blood cells from destroying unintended brain structures once inside the central part of the brain? Thank you for watching, and we look forward to potentially pursuing this idea.
At first we just kind of began with thinking about what medical problems we wanted to try to fix, and one of the ones we thought of was Alzheimer's because, well, right now there isn't really any cures for Alzheimer's, and we wanted to see if we could try to find a possible solution because Alzheimer's is kind of a problem. It's because there isn't really a treatment for it right now, and it would be nice to be able to have one.
I don't have any specific questions right now, but I love the creativity and the fact that you're inspired both by Alzheimer's, which is a big problem. There's a lot of unmet need, and you're young enough now to start trying a cure before you actually need it, which is great. And I also, for the first group, really appreciate looking at creative ways to handle things in the environment, because it's not a thing long term that you'll be impacted by. And I think the microplastics is a huge one because they're everywhere. So I think that I love the creativity. I love the passion. I think with respect to getting stuff across the blood-brain barrier, one of the things you can look at is looking at how gene therapy is going to be delivered, which is actually injecting directly into the brain. So I would look at some of those resources to see if there's any routes of administration that might be possible for you.